Hello and welcome to the Elements of Deep Sea Mining. I'm Eric Young. Finally, a marine biologist. Dr. Jesse van der Grient is a quantitative ecologist who specializes in how ecosystems connect, how species migrate, and how stressors interact in unexpected ways.
Imagine you're going to buy a new car. We can start with different levels of information about the car. I can tell you that the car has roughly 40,000 parts, just like any other car. I can tell you about the numbers of each unique part and their sizes or weights. You probably want to buy the car after you've known something about how does it work. Can it drive? Can it brake? Does it look nice? I mean, ecosystems don't need to look nice, but you get what I'm saying. I'm more the quantitative marine ecologist. I often describe myself as an ecosystem scientist. I want to know roughly what does the food web look like, how does it function, what is it structured like, because that can help me then start looking at impact.
Jesse works with the Deep Ocean Stewardship Initiative, a group of experts who don't advocate for or against deep sea mining, but participate to inform decision makers so that whatever decisions get made are based on sound scientific foundations. Without further ado, I bring you my conversation with Dr. Jesse van der Grient. Thanks for listening to part one of... This was a long conversation, so we've split it into two parts. Tune in next week where we get into some of the specific risks and the impacts. Okay, welcome, Jesse. Thanks for being here.
Thank you for having me.
So Jesse is a marine biologist and is one of the most recommended guests to the most recommended guests to the podcast. And
wow, thank you.
Yeah, well, I mean, don't thank me. I didn't recommend you. I just asked for who I should have on to discuss the kind of potential impacts and the things that people ought to know about the ecosystems and the biology that we're talking about when we talk about deep sea mining. And so your name was put forward. And yeah, I'm just hoping that we can kind of put some nuance to the picture. I think everybody who listens to this podcast is kind of aware that deep sea mining involves trade-offs, and one of the main trade-offs is impacts to the natural environment. But I think a lot of people are kind of wondering what the specific issues there are. So I'm hoping that we can get into that.
Sounds like a great idea. Yeah.
So let's just start with your kind of like main message, a main thing that you'd like a listener to take away from this conversation.
Yeah. No, that sounds good. So my main message is that we need to understand what is being managed. So that includes so many different variation types. Like we have different habitats, both in the abyssal plains themselves as well as seamounts. We have variation in space. We have variation in time. But also we have a lot of connections in the deep ocean that people don't always know about or understand very well. So this can come from migration. For example, long distance migrating animals, those are the better known ones. But there's also a lot of migration via ontogenetic migration, which is kind of different life stages using different habitats. So we get all these wonderful connections in space and time via species moving and using different habitats in their lifetime. So we need to have a better understanding of those kind of different types of sources of variation. And we need to incorporate that in management in order to manage our resources well. That's not just deep sea mining, that's all sorts of resources.
Well, so I like to say that I'm someone who falls into the broadly kind of systems thinking, systems theory bucket in my research. And so I really like this idea of we need to not only focus on the things themselves, but the interrelations and connections between all the things in order to understand the picture.
Yeah, I'm glad we're on the same line then. That's my biggest interest as well in all the research that I do.
Well, so then take us into you, how you got to be working on this topic, but maybe you could start a little bit farther back than that. Where do you come from, and how did you get to be a scientist in the first place?
All right, yeah. So I would describe myself as a quantitative marine biologist, and I work both in shallow waters, coastal environments, as well as the deep ocean. I'm from the Netherlands, but I studied in the UK. I did a bachelor degree in biological sciences at the University of Oxford. I will admit I was interested in so many different things. I did know I wanted to go further into research, but I was like doubting between the deep ocean, plant development, social systems in ants. Like it was quite varied. But eventually I was like, no, I was really interested in the ocean. And I had this great opportunity to do a PhD in the deep sea. So that's where I went to. And that actually hooked me so much that I never left. So during my PhD was also at University of Oxford, and I looked at macroecological patterns in bristleworms. So macroecological patterns are like these large scales patterns that we can see, looking at, like, for example, species diversity, body size. And I used the bristleworms not because I'm particularly interested in worms, but more because there's quite a lot of numbers for them. So this is where my love for the more mathematically modeling side came up. So after, like, when I was looking at the worms, I was also looking at short and long-term environmental change and fishing impacts on the worm communities, the polychaete communities. But then I moved a whole 100 meters to a different department and actually on ecosystem modeling. So I was working on this new type of kind of like a connection interaction network model, but we were modeling cumulative impacts. So we were interested in many different simultaneously impacts that happen in marine environments. We were focusing on the California current system as well as the Chukchi Sea. And me and my supervisor, we isolate, like quite often what you see in cumulative impact models is that they have multiple stressors, but they're either additive, so they're added to each other, and that is it, or they might just do a multiplication between the two. But that's not often what we see. And there's already some data where you can isolate the interactive effect from the single effect. So that's what this model did. So we're really going a little bit deeply into that.
Okay, so explain that just a little bit more for me. So I can understand, that if you get multiple different stressors, it's intuitive
Yes.
that you just kind of add them up, and then I can sort of understand that if you're already suffering from one stressor, having another stressor is going to be kind of more than just additive. So you could multiply that. But what other kind of interactions are you talking about?
So that's a really great question because I should have explained that more. So when you have two different stresses, yes, they can add up and then you can easily predict the effect if you have both stresses there, but they can also interact and then they can turn into antagonistic or synergistic effects. So antagonistic is that where you have, if you had summed the effects up, you might get a certain level, but by having both of them, one is canceling out the other a little bit. So then you get an antagonistic effect.
Maybe we should clarify that stressors in this context means kind of any sort of pollutant or impacting change to the environment, because I think that's a bit that's a bit jargony for people who study.
That's a good point. Yeah, so any kind of like effect or impact that we have, there's a lot of words going around. I've seen pressure, impact, effect, stressor. Doesn't really help.
Yes, so this can be things like whatever, like pollutants or temperature change, climate change.
Yes, fishing is an impact, drilling, dredging, mining, that will be an impact.
Right.
But, introduction of new species, invasive species, that can also be an impact. So there's all sorts of things that you think like, okay, if a system was healthy and kind of closed, so no new things necessarily coming in, then that anything that changes in that could be an impact. So that can be, it's quite often an out. So I'm going to go into technical, aren't I? I should use simple language.
No, that's okay. Technical is allowed. I don't think it's hard for people to follow.
Something new, something different, something probably non-natural.
It is a bit surprising that you would get this, what did you call it, antagonistic relationship where stressors can cancel each other out, because I just assume that normally any kind of change is a stressor. So you would think that two changes just make for a bigger change.
There are definitely combinations out there where you actually see that cancellation impact, but more often we see actually that they kind of make the effect a lot bigger. So you have additive effect and then a little more. There's also, it's hard to do these experiments, and having more than two stresses is even harder. But there have been experiments done, and the indications are that if you have more than two stresses, that likelihood of getting synergistic impacts increases. So I'm not surprised that you are more familiar with the synergistic effects, because there's kind of an assumption the more impacts are around, the more likely things will be synergistic.
And how hard is it to do that? So like you just sort of mentioned, mostly people try to do one stressor at a time.
Yes.
Because once you add multiple variables, then it's just really hard to actually separate the signal from the noise.
Yes, so it's hard to like do these experiments. So when we're doing experiment where we actually testing for the effect of a particular stressor, then you first need to think about like what animal or organism do I have? What's the best setup to keep that animal happy? Because you don't want it to be so stressed that the impacts you're measuring are actually from the stress. So you need to make sure that it's in a good environment where if there is a change or an effect, that is from the stressor. You need to think about how you're going to apply the stressor. So, for example, if I did an experiment with ocean warming, am I going to increase the temperature in the tank at a certain rate? Is that rate going to affect the responses? Am I going to increase a little bit and then just let them adapt, and then increase a little bit and adapt, and then hold it there? Is it going to be a male-female difference? Is it going to be a life stage difference? So there's all sorts of things that we need to think about before we've done the experiment. We need to make sure that we have enough animals that we can pick up on signals. We need to decide what we're going to measure, because we can measure behavioral changes, we can measure respiration changes, we can measure like protein or gene changes. So there's a whole lot of things that can happen in that, and that's just for one stressor.
Right.
Once you like have kind of verified, but like this is what that one stressor does, you can introduce a second one, but you ideally also know what that single stressor does on its own, so that when you combine them, you can start teasing apart what's that interaction effect. Is it getting worse or is it getting less than expected based on the effects of those two single stressors? And then you need to do a third.
Yeah. So this
Well, species quite often. So you'll have to do a lot of different species.
Right. So the complexity here just explodes as you try to add additional variables.
Yeah. Unless you know what could be like a good indicator species that is perhaps more sensitive. So if you notice like, hey, this organism is gonna, be negative and very strongly negatively impacted at a certain level of temperature or acidification or oxygen loss or sediment plume, then, if we monitor in the wild, we can then see like, oh, this animal is now no longer happy. The rest is still fine, but this is our indicator. This is our early warning signal. It's like this traffic light starts turning on orange. This is the time to make changes. But we quite know what the indicators are.
Well, I want to ask you about what the indicator species are for some of these deep sea environments. But before we get there, I interrupted you taking us through your kind of research career. So
yes,
how did you get from doing ecological modeling to specifically focusing on deep sea?
Yes. So after that kind of the cumulative impact modeling at Oxford, I moved to the University of Hawaii. And there I actually started my deep sea mining career, if you like. So this was on a project looking at the potential deep pelagic impacts. So quite often with deep sea mining, it's been a real strong focus on seafloor impacts, and that makes absolute sense because that's where the mining vehicles will be. It's where the plumes from the vehicles will be created. But in a lot of the designs from the vehicles that we see is that when they're bringing the nodules or crust or polymetallic sulfides, when they bring it up, there will be some unwanted material. In the abyssal plains, it will definitely be sediments, but there's also a lot of water that will be brought up, and that's going to be discharged back into the water column. And the question is like, well, if it's released at a certain depth, can there then be additional impacts? So I thought there would be some more data when I started, but it was actually very little because the idea was that I was going to do some ecosystem modeling, but there just wasn't that much data yet. This ecosystem model is going to start. I know there's been a different version also from someone else, but I'm going to make my own one with data that we've collected in the CCZ. But in this project for the University of Hawaii, I then pivoted a little bit and I was thinking more about ecosystem services. And there I started looking at tuna fisheries that were overlapping with deep sea mining areas in international waters. And it was just the first principal exercise being like, if these two industries occur in the same area, could there be an issue? So the question then would be, is like, well, are there being tunas caught in these areas? And if so, how much and what kind of countries really catch the tunas there? And there were some really interesting differences. Some countries were fishing a lot in certain areas, especially in the Clarion-Clipperton zone. There were real big differences between countries. So then, you can start asking questions about equality issues, like if tuna fisheries are being impacted, who then carries the cost? And yes, sorry.
So that sounds like, that sounds like actually quite a pivot from like hardcore quantitative biological modeling to
Yeah,
going much closer
there were
down
numbers
the
involved.
social sort of.
I mean, I didn't delve very deeply into the potential inequality issue. Like that's not my field of expertise. But like just looking at the two different spatial overlaps, that is the first principle one. And it offers opportunities to then talk to funders, being like, hey, I want to make this ecosystem model. There could be an impact. It could
Yeah.
be useful to think about trade-offs between industries, but we need to know if there's going to be an impact. If the plume is released too shallow, let's model this.
Can you define ecosystem services?
Yeah, so that's I didn't even explain that yet. So when we're thinking about the deep pelagic, there's a lot of different zones. So you go from the surface all the way to the seafloor. And
Hmm.
over 200 meters, this is the sunlit zone or epipelagic zone. And between 200 and 1,000 meters, you've got the mesopelagic or the midnight zone. And especially in that zone, you have these diurnal vertical migrators. So these are squid, fishes, crustaceans, and they swim up and down the water column every night. So they come up the surface at night, feed there, and then swim down just before sunrise. That is, first of all, an important carbon drawdown. So there could be like a lot of carbon moved that way to deeper oceans where it could be sequestered because they poop in deeper waters. But these animals are also really important prey for tunas. So I'm more interested in that fishery support ecosystem service where the diurnal vertical migrators are the prey for the tuna. So if the plume is released in that mesopelagic zone and the diurnal vertical migrators are negatively impacted to a point that it's going to affect the tunas, then that will be it. Then you can start
Yeah.
monitoring the loss in the tuna fisheries. If the plume is released deeper, then that would be one problem in a way solved.
Yeah. So it's kind of this kind of work that influenced the companies to change their models from releasing in less than a kilometer to, I think, deeper than 2 kilometers. A lot of them have said, oh, well, we heard the scientists say deeper than 2 kilometers, so let's just go deeper than
Yeah.
2 kilometers.
I hope it has played a role. So with the team in Hawaii and with I would say the metals company, we actually did a lot of baseline studies in the Clarion-Clipperton zone, where we did find a lot of like really interesting patterns that we just didn't expect yet. I'm not going to say anything. The papers are coming out, so it's not my news to share. But there were just some paradigms that were being challenged. So that I think also played a role because very quickly after those results were being made to the contractor and a few other things, they started to kind of talk about deeper and deeper. It's only the metals company that has said we're going to release deeper than 2,000 meters. And at the same time, it's like it's nowhere in the regulations that they have to. So, if it becomes technically or financially or logistically challenging to do it that deep, because I don't actually know if they tested their system at 2,000 meters. I think they tested it at 1,000 or 1,200 meters. So
Okay.
I don't know if that could change then. But that's a regulatory question almost. As a scientist, I can more highlight some of the issues that might come by releasing the plume shallower than 2,000 meters.
Yeah. Well, I guess that's interesting. Maybe do that like specifically because this is a little bit why I dragged you on this podcast is to say, okay, yeah, we don't want to release a plume into the area of the water column where there is kind of active marine activity, especially the kind that is going to filter into the food chain of tuna and species that humans rely on. But yeah, what does that mean? Like, what are we actually worried about with releasing sediment into that area?
Yeah, so I've actually, when I was at Hawaii, I also did a lot of work on suspended sediments, looking at, well, first trying to see if there was any deep sea data on deep sea animal responses to suspended sediment increases. There is a little bit. There is currently enough to start a meta-analysis that is not out yet. But at the time, that also kind of shows how much suddenly we have output there. So that's good that more people are trying to do those experiments. But at the time, we basically had shallow water data. And just to kind of give a range, like if you have a sediment plume, there's a lot of responses that can be measured. Kind of this is also tying back into that chat we had about the experiment with ocean warming. So if you have sediments, I mean, you can have behavioral changes. So some animals might not realize that they can't see so well anymore. So they start to explore around a lot more and then actually have an increased risk of running into predators. They might actually be hiding a lot more. We see in a lot of shoaling fish like guppies that the shoaling cohesion gets lost and then they're also more vulnerable to predators. We see changes in feeding interactions. So predators, although they have a higher risk to run into those little prey that get confused and just start to be brave, they can't see as well. So they might actually not have as much time to respond to a prey. So that chance of actually catching your prey reduces. You might see sediment particles harming gills or soft tissues. We see a lot of filter feeders either rejecting sediment particles, but therefore also food particles. So they will starve or they just eat sediment particles and then feel full, but they're actually not full because they don't have any nutrition. So they starve. We see a lot of changes in respiratory procedures. So sometimes a lot more increases in respiration, like stress. Like if you get a little bit excited or run or fear, you actually start to increase your heart rate and breathing rate as well. But in others, it will actually reduce. So there's a little bit complicated what happens there. We see an increase in stress hormones. In longer term studies, we see that the changes in reproductive output. Quite often reproduction goes down. If gametes are released in a plume, then actually sediment particles will get attached to the egg or sperms cells and then they can't find each other anymore or they just sink to the bottom. Or if there is a fertilized egg with sediment particles on there, you get abnormal development of embryos. We see increased mortality. There's a whole range of things that happen just by releasing a plume. Now, the difficulty becomes with how much sediment elevation, like how much sediment in the water actually creates those problems. That varies. It depends actually to the length of exposure. And you can almost imagine this as like if I made you run into a very heavily smoky room, just run through it, you'll be fine. But if you have to stay in there, you're not fine for very long. Now, if I put you in a room with just a little bit of smoke, you're going to feel it eventually, but it will take a much longer time. So that time and magnitude have a really interesting kind of relationship that we still haven't very well developed. So the longer the exposure, the lower the threshold. And we actually start to have impacts, unless it's overwhelmingly bad. But eventually, you need to think about that exposure length. And then there's also the question about how often does it happen. So with the plumes, what we can kind of see sometimes, not in all cases, but sometimes if there's a recovery period and the animal has maybe time to clean itself or take a couple of good breaths, then the next exposure will be as if it was the first one and it's fine. But others might need more time to recover. And then because they just get repeatedly hammered, then they go down. So there's lots of different questions. So that's just the plume in the shallow waters. There are some indications that it might also play into different ways in the deep pelagic. Because in the deep pelagic, in that mesopelagic zone, a lot of animals rely on communicating with light, bioluminescence. And for that, you need clear waters because light signal, it only goes so far. So if you add a lot of sediment particles in the water, then that light signal, that distance becomes smaller. Now, maybe for prey, that's nice because the predator can't see them anymore, but they also use that light to communicate to each other. The problem is that most of the time, light is actually used as a camouflage. So you have in the mesopelagic, you have down-dwelling light, and because it's already so deep in the water, it's literally straight. So what we see is that a lot of these animals, especially diurnal vertical migrators, have these photophores, these organs where they can emit light with, on their belly, so that they can match the down-dwelling light. In order to do that, the light needs to be straight. Now, if you put a lot of particles in the water, that will scatter the light. So suddenly those photophores, that bioluminescence on the side of the belly, is not going to like camouflage them anymore. So they will actually throw shadows from all sorts of sides. So they will become more visible to predators. And that kind of changes in turbidity have been shown in like streams. So it's very likely that it will also happen in the mesopelagic.
So these animals have lights on their belly so that if they're essentially if they swim above a predator, they don't
Yeah.
show up as a shadow. They just show up as this kind of same amount of light
Yeah.
that's so interesting. I
Yeah.
had no idea. And so this deep pelagic that you're talking about where there's
So the deep
more of this.
pelagic is anything deeper than 200 meters. And then the
Okay.
mesopelagic is part of that. So it's the 200 to 1,000, 2,000 meters. That's quite a well-studied area for the deep pelagic. But deeper down, there's all sorts of cool animals that we haven't studied as well. And there are zones, but there's some discussion about how clearly defined are those zones.
Can you talk a little bit about the lack of data here? I thought that was very interesting. You said you went to Hawaii to do one analysis, but you got there and realized there just wasn't the data for that, and so you then had to pivot.
I thought there wasn't enough, like, because there's always some data, right? But is it enough to inform your models, and is it good enough quality?
Yeah.
So for my ecosystem model, what I didn't want a simple ecosystem model, because if you make the ecosystem model, the food web, too simple, then you're not really learning anything from it. It needs to be at least region or area specific and capturing the main patterns or functions that you see. So for me, that would be like, well, what kind of species groups do we have? Like functional groups. What kind of predators are there? Suspension feeders? Are there differences in like trophic interaction? So you can have two different predators, but one can be like an ambush predator, one can be like one that actively hunts. You need to kind of capture those things in your model, especially if you're thinking that their responses to this, for example, the sediment plumes might be different. Because if you model them as one whole, you can't build in those nuances, so you can't actually calculate what the impacts are. So you need to understand that bit. You need to understand what animals are migrating and from what depth and how often. Are there anything like environmental variables that correlate with it? So for example, one study that was using bioacoustics really showed the influence of the oxygen minimum zone in the clarion-clipperton zone. So this oxygen minimum zone is an area with extremely low oxygen concentrations, and that kind of, it's not a barrier to diurnal vertical migrators, but it did influence how deep they were migrating. So if the zone was if this oxygen minimum zone was a bit shallower, then the diurnal vertical migrators were going a little bit shallower during the day. And more of that community was migrating during the night. So you have this like larger proportions migrating, as well as being somewhat shallower compared to the areas where there might be a little bit more oxygen, or the zone, like the really core areas were a bit deeper. So it's quite important to know those parts, and we know them now, but we didn't know them at the time I started. So
this is fascinating. So what you're saying is you need to have your model be sort of complicated enough so that it's interesting or relevant or something.
yeah,
But there must also be a point, like we were talking about earlier, you get too many variables, you can no longer just, so it must be a bit of an art to tune your model with the right amount of complexity without making it so complex that you can't tell whether or not you're.
I've never heard it be described as an art. I would always see it as a game. You want it realistic enough, specific enough, but not too complicated, because we have other biologists to really go into the details of species-specific things. But for these ecosystem models, I mean, there's some redundancy in any kind of ecosystem that's healthy enough between species functional roles in the ecosystem. So you're really focusing on those roles rather than the specific species. But if there are some really big differences between the species in the same functional role, then you need to start thinking about, hey, should I split them or not? So as an example, something I did in the Falkland Islands, obviously that ecosystem model needed penguins in there, but there were two species of penguins that were foraging quite close to the coast, and one species was foraging much further from the coast. So it's natural already looking for different species different food items. So they're both penguins. They kind of have the same ecological role, but the one that goes further away was much more looking for like lanternfish and squid, and the ones closer to the coast were much more looking for like lobster, krill, and that sort of thing. So those kind of like they were in the same functional group, penguins doing their own deep, like big fish niche role, but because there was such a big difference in what they were eating and where they were eating, I split those. But the two species that were doing very similar things, they were kept together because that was just big complicated otherwise.
Well, okay, so then, fast forward, how do you get from there to now?
Okay, yes. So in Hawaii, after my sediment plume work, I actually got involved with the Deep Ocean Stewardship Initiative. So DOSI is a network of experts, scientists, lawyers, work with engineers, policymakers, industry, and we do different things. But I focused mostly on the minerals, on the deep sea mining. So there, I still kind of kept my connection to deep sea mining as part of DOSI. DOSI has observer status at the International Seabed Authority, so we go to the meetings, present latest science. We are ready to help with any kind of scientific evaluation. Quite often put in comments on environmental impact statements and looking at regulations from an environmental perspective. And that kind of kept me in the loop because after Hawaii, I moved to the Falkland Islands where I was much more looking at coastal areas, squid fisheries. So no deep sea mining. And right now I'm actually back in the Netherlands looking again at cumulative impacts, this time in the Wadden Sea. So can't actually get shallower than the Wadden Sea. But again, looking at cumulative impacts and thinking more about what kind of frameworks can we establish to make that more quantitative assessment? Because in a lot of cumulative impact assessments, when we're looking at the sensitivity of organisms to these different impacts, it's more qualitative, informed by expert judgment. Okay. There is a lot of data, especially for the Wadden Sea, but kind of wrangling that into more of a how do you use that then for this kind of assessments? That's what I'm working on.
Interesting. You still, under DOSI, you still attend ISA meetings, you still keep one foot in the deep sea conversation?
Yeah, so during day, I'm shallow, and then during the night, I'm a deeper.
Yeah, so it's kind of the opposite of the migratory species.
Yeah, a little bit. Yeah. But it's really, I mean, there's definitely lessons to be learned from studying these different kind of ecosystems, because in the end, I am interested in the structure, functioning of ecosystems, how impacts can change the food webs, and things you can learn in one area can potentially be applied to other areas. So the deep sea, quite often data limited. The Wadden Sea is quite data rich, but has its own challenges. So kind of study them both in a similar sense. You can kind of see like what is similar, what is not similar, what kind of framework can be used in both situations. Where do I need to change it? And if I know I need to change it because of this data dependency, I can make my work also more general for other areas.
Yeah, it's interesting this this move to get away from kind of expert reliance on cumulative impacts and interactions. I'm tempted to ask you more about that, but I guess
I mean, I will just say, like, because we have, science is not standing still. We're doing a lot of work, and lots of different groups kind of focus on different areas. So you might have people focusing on climate change, focusing on parasites, focusing on fisheries. And I'm the kind of person that tries to kind of talk to everyone and combine that data. So,
yeah,
like before, we didn't always have the opportunity to look at it more in a quantitative setting, but now we are getting there with our better statistical models. It actually starts to become possible. So that makes
I think it'd be very interesting to do that. I mean, you at a certain point you would kind of start to have the models show that the experts are wrong in some places. I think that would be an interesting place, because I'm always like a little bit, I wonder about this, like, let's get all the experts into a room and then ask them about these really complicated questions about interactions. Experts have, kind of mental models that they've built up that are quite accurate. And then at some points, and imagine they limit out
Yeah, I think the biggest change is what we're going to see is like, if you look at the qualitative frameworks, they have to score it between like just a limited number of, groups or 5 or 6 groups. It's hard to kind of capture really big changes in if you have only like 5 or 6 categories. And
Hmm.
more often than not, that misses the non-linear aspects that can happen. So we kind of always assume that things happen in a linear scale, but what we're finding more and more is that things are not linear. So
Well, that makes sense given, I mean, that also makes sense just given how bad human cognition is at nonlinear thinking. We're really bad at seeing how exponential changes
yeah,
can really make
good
effects.
point. Yeah,
It's unintuitive.
yeah, yeah,
Okay, so then I think we can jump to kind of the reason that you're here, which is to give people, um, yeah, just and kind of a 101 intro to what are the vulnerabilities of um, deep sea ecosystems to deep sea mining. And I guess backing up a little before that is just kind of like what are the ecosystems of the deep sea mining areas like?
yeah. That's a good question. Yeah, because we kind of always treat there's different areas that are of mining interest, and they fall into like 3 broad categories. You've got your polymetallic nodules on abyssal plains, and you've got your cobalt-rich ferromanganese crusts on seamounts. So these are like these rich metal layers that can be formed around seamounts, and then in certain hydrothermal vents underwater volcanoes, you have these polymetallic sulfides, and they I even say this like, oh, you find this here, this there, but there's already kind of an indication that in some cases they overlap. So they're not always as nicely delineated. So, for example, in a lot of areas where you have a lot of seamounts, you might also find nodules. You might find the hydrothermal vents. So that makes it a bit more complicated. And I will admit, we are only really starting to be aware of that ourselves as the scientists. So there's hopefully a paper coming out very soon that kind of details that more and makes the case. But in the mining regulations, we very much treat them as if there's just one thing. And
Okay.
then with the abyssal plains is a very interesting one because for a long time we just kind of assumed that they were homogeneous, but they're actually not. And there's a lot of variation that we maybe as humans didn't appreciate. But the biology does. We see a lot of changes like this can be dependent on the nodule abundance or the size of the nodules, or whether there's like ripples in the sediment or troughs or something like that, or whether there's a hill or not, or something bigger than a hill but not yet a mountain. And that can really influence where food is going to be accumulating, how currents, even in the deep ocean, you have these currents slowly, very slowly, but there are currents that can really transport things. It can influence connectivity. So I don't know, when I started my PhD, I was learning about the abyssal plains. It was very much like abyssal plains are homogeneous. You'll find the species all over the place. But now that we have more data from the CCZ, we can actually see that things vary and can vary quite rapidly. So in certain contract areas, if you study the eastern part compared to the western part, you see big differences. It can be contract areas that are right next to each other that are very different. This can be easily influenced again by these like large scale features of the oxygen minimum zone, if it's present above it or not, or seamounts or currents that are going to the system. So there's a lot more variation, and we see that in the species that are present there as well. We quite often find singletons or doubletons. So you find a species, but only like 1 or 2 individuals of that. And then you might not find it again, or maybe in a different area. And then also these really low abundances. And it makes you wonder, how can these species persist if they appear
Yeah.
in so low abundances? And how do they find each other to mate? Or do they just release their eggs and sperm in the water column and hope for the best? I mean, this is not my area of expertise, the like reproductive biology. I know there's like different strategies, but it's not necessarily my expertise. And a few studies have been looking at connectivity, but it's still very hard to actually determine if there is connectivity in, for example, the CCZ, and what that is and how that varies between different species. You do need a lot of like numbers for that, because in order to look at connectivity, you kind of need to look at the genetic material of animals. But if you only have 1 or 2 individuals from one location, you're not going to know anything. So you need to have a few individuals from different areas before you can really see are they connected populations or not. So that makes it a bit hard.
Say a bit more about what connectivity really means here.
Yeah, so connectivity, you can see it as like when we want a population to persist, they obviously need to meet each other and they need to mate so that they can make new little babies, and that needs to continue. If you have a group of animals and they all intermingle, then there's a really nice mix and everyone has the kind of same genetic material. You can see that in something that's called haplotypes. So in your mitochondrial DNA, which are like your little energy machines in the cells, they have specific DNA. You can really look into like quick changes in that DNA compared to your more cell nuclear DNA, which is much more protected by proteins and stuff. So you can look for specific changes. And if the population mixes really well, then everyone will have kind of the same type of these haplotypes. So you don't really see many changes. But if you have a group and it doesn't mix very well, and things on one end mate with only those and things on the other end mate with only those, you can maybe see a random change happening in one, but not in the other. And you can start seeing that in different haplotypes. So then you see that connectivity, there might be still some exchange, but much less. And that also means that if there is something happening to that population, if half of it gets eliminated, then that has consequences for how quickly that population can come back. Because here you only have half of them breeding, while in the other one you had the whole thing, it was all the same and it could spread quite easily.
Right. So instead of having whatever 1,000 individuals because they're all the same species, you think, well, they actually maybe are two groups of 500 sort of thing.
Yeah. So that has consequences for how easily those populations can disappear or not, or can recover or not, or can like come back after a disturbance. So we do want to know that connectivity. If it turns out that you have these really restricted populations, that really changes the conservation and management question compared to everything being connected.
In terms of having an overview of the actual populations in these areas, like, I mean, how is that when you say that you found kind of like a couple of individuals and you're not sure how many more are out there? I think, yeah, well, that makes sense because it's dark and kilometers away from the surface and you can only get down there, rarely with a limited amount of spatial coverage. So it's like, okay, yeah, we find a couple of things, then we have to guess about what else is there, or do you have vehicles kind of roaming and mapping and you actually get a good population census? Can you say more?
Yeah, that's a really good question. So there's different techniques for different kind of organisms. So when you have the ROV or the remotely operated vehicle down, get the footage, you see things that are big, like corals, like sponges, like fishes. But there's a lot of small things out there, like, for example, those bristleworms that I did my PhD on. You can't actually see most of those with a camera. So in that case, you take a sediment core, like a box core. It's literally just a box that you can drop to the ocean floor, and it takes a scoop of sand, and then you can sift through the sand and then find your crustaceans, you find your mollusks, you find your polychaetes and other worms. And then there's even things that are even smaller. So you might need a different core that is more round, like a megacore, so that goes deeper in. Because they're smaller, you don't need as much surface area, but it's much more interesting to look at it also from a depth perspective. So there's these different techniques that you need. So one technique doesn't catch all. If you want to look at the swimming stuff, the pelagic stuff, then none of this might actually work, because if there's an RFE, we know that some of the fish or the squid actually get scared by it, so they'll move away. Some also get attracted by it, so they will come in and you get these really weird kind of ideas of what is in the community. For some of the communities, like the scavengers, you can drop a bait with a camera, with a photographic camera, to see what is being attracted over time. But there's always these questions like, how do you actually know the community? But by trying these different sampling techniques and in different locations, maybe different times of the year, across different years, you can start building up a picture of like, how likely is it to encounter a new thing? So that can already help you, because the idea is that when you start taking samples, the number of new things kind of rapidly increases, but then it actually starts slowing down. Yeah. And those kind of curves can help you predict how many new species you would expect. We're never going to find all the new species, but we can start predicting like, hey, how far are we off? Do we know half of it? Do we know 90% of it? And that gives us a better idea of like, what's the species richness, what is a community in an area like, and then we can start comparing it more to other areas as well. The one difficulty that we're starting to have is, but now that we have more material and we're doing these genetic analyses, we actually do find out that things that were thought of being the same because they look the same might not be the same. So that becomes a whole different Pandora's box. I'm not a geneticist, so I'm not going to go into detail in that, but it's just another thing. Like, it's a little bit more complicated than we thought. We thought there were species that were ranging across the ZZZ. Turns out it's a species complex of things that look very similar, but it isn't.
Yeah, interesting. I guess that makes a lot of sense when they're when they're types of species that we don't have a lot of experience with, we wouldn't know exactly. Like think about, I don't know, bears, for example. There's bears range all over the place. Some of them are in the same species, even though they look very different from one another, and some of them can be in different species, even though they look kind of similar to one another.
Yeah. Also in butterflies and beetles, we know these are very common problems. They're not just in the deep ocean, but you kind of want to know roughly what is out there before we do any kind of exploitation work.
So then this is maybe an unfair question, but can you put the amount of uncertainty about the deep sea environment into context of other environments? So people love to compare, the deep ocean potential mining areas to the rainforest and say, well, there's such and such many species. But I wonder, kind of, even if you compare it to, I mean, I don't know what a fair comparison, but maybe to other ocean zones and things like that would be interesting to give people an idea.
I mean, I'm first gonna get just rid of this this area versus like this habitat versus that area. I don't think you can compare deep sea habitats to rainforest. One has trees, one has fish. That's just us gonna start discussing whether Amsterdam is a better city than London. It's like different kind of aspects to it. They're not comparable. So I don't think it's a fair comparison. By having these comparisons out there in the discussion, it just muddles the water. It doesn't help us focus on what is going on with deep sea mining, because whatever is happening with deep sea mining, I don't expect it to have big changes in terrestrial mining because there are different companies, different countries quite often, different people involved. Whatever is going to happen in deep sea mining, I don't think it's going to change anything terrestrial mining. But when we're focused on deep sea mining, there's lots of different types of uncertainty, and it's difficult to kind of decide like what, I don't know, how can we know what you had a way of phrasing it, how do we put it in context? That's a hard question, and it will really depend on who you talk to. So I think there's lots of different ways of bringing in uncertainty. Uncertainty comes in what are we studying? How are we studying it? We just mentioned the different sampling techniques for the communities. We haven't even talked about how they're interacting. And we don't need to know everything. We just need to know enough. And that question of sufficient knowledge, that's still a little bit unknown. And I've been trying to think like how, because I've had these discussions like, oh, we know the numbers, we know the biomass, like we know the system. And I would argue we don't know the system if you just know the numbers. And I thought like maybe if we talk about it in like a car, imagine you're going to buy a new car. Probably want to know something about that car rather than just a car. I mean, like I'm really focused on colors of cars, but even I wouldn't agree to like, oh, this is a red car, just accept it. I want to know more. So we can start with different levels of information about the car. I can tell you that the car has roughly 40,000 parts, just like any other car. But that's not going to convince you to buy that car. I can tell you about the different kinds of parts that are in the car, each unique part. That's not going to make you buy the car. I can tell you about the numbers of each unique part and their sizes or weights. That's not going to make you buy the car. You probably want to buy the car after you've known something about how does it work. Can it drive? Can it brake? Does it have all the safety regulations in place or not? Does it look nice? I mean, ecosystems don't need to look nice, but you get what I'm saying. Like, you want to know a certain level. And where that level is will vary for different people. I probably need to know less about this car than my dad. Whether that's a good thing or not, I don't know. But that's, so it will really depend on who you're asking. I'm more the quantitative marine ecologist. I often describe myself as an ecosystem scientist. I want to know roughly what does the food web look like, how does it function, what is it structured like, because that can help me then start looking at impact. But other people might be really interested in a particular group of species that they're really focused on, they've been studying their whole lives. Other people might be more interested in not the biology, but the genetic resources, new information that, I don't know, pharmaceuticals can use, or they can use as an innovative biomolecule, or others might be more interested in like the impacts on fisheries. Like, what is enough? It's really hard to say. There is, however, definitely ways to reduce uncertainty, and that's by taking samples. So if we're taking samples, the more samples we have, the better we estimate the average, and the more likely is that these error bars, uncertainty around an average, will go down. The complicated thing with that is it will depend on the area, it will depend on the species that you sample, it might depend on the season, it might depend on the year, and all of those things bring different uncertainty. So this is kind of where we have these broader discussions about how to sample the deep ocean, how to use that information for things like environmental impact assessments. The International Seabed Authority, the ISA, has already set out different documents, what is needed information-wise for environmental impact assessment, so we know what type of data we need. There's discussion still ongoing about what kind of sampling regime or like certainty do you need, because if you go out and you know there's probably already a difference. So for example, if you're going to sample the sediment versus the nodules, you can probably expect a difference in species richness or abundance or biomass. But if you just take a few samples, they will vary quite a lot, and therefore they can overlap. But you can do power analysis, and then you can do the power analysis before you start sampling to know what kind of sample size do I need to start actually picking up on those signals. So there's many different ways that we can talk about uncertainty. There's many different ways of how to reduce uncertainty. In the end, it is coming down to how many samples do we take, what do we do with the samples, how are the samples taken, and how are they going to be used.
Yeah, and what is the kind of the goal, the purpose, the value that we are interested in? I really like your car analogy. I think that was super helpful in terms of putting the numbers into context. I was expecting you to compare this to some other ocean environments.
I'm doing my best there, but no, yeah, I mean, because
No, I think that was a great bit of context to put in, and it struck me also that yeah, people, like you said, people are interested in different things about the ecosystem, and in the same way that people are interested in different things about the car. So if someone is interested in bioprospecting for species that might lead to new healthcare drugs, then they're going to be interested in kind of the properties of individual species and just not losing access to those species.
yeah. So I think that uncertainty, quite often that aspect gets lost in the uncertainty debate, because they always say like, oh, scientists just want more samples, which is true. We always want more samples. But at the same time, we don't always talk about like how are the samples used. Like what, if we just look at abundance, just look at biomass, just look at species richness, that's not enough to manage an ecosystem. That's not enough to buy a car on. It's not enough to manage that ecosystem. So we just need to have that little bit more. And the ISA regulations do ask for that little bit of more. But I think it is a little bit different from how industries used to start operating. So this is the first time really where we write the rules before the industry happens, right? So that makes it really interesting. And there's been some really big changes in how we manage our ocean environments. And I think that hasn't always trickled through to things like oil and gas. So for example, in fisheries, more often now you see people thinking about ecosystem-based fisheries management. So they're not just looking at, hey, what's this fish, how many individuals, or what's their biomass, but they're also thinking about like, how does it reproduce? When does it reproduce? Does it need specific habitats for that? Does it need specific prey for that? Does it need different prey when it's younger compared to when it's older? Are there specific environmental influences that can really influence the success of recruitment? And we're also starting to build that out a lot more to these, like not just the prey, but also predators on those target species. And in the ISA, there's also this statement that they want to go more towards ecosystem-based management. So ecosystem fisheries management is really just looking still at the food webs and only the fishing. But if you also start considering other industries, then you're talking about ecosystem-based management. So it's recognizing that there's different uses for our oceans, people with different objectives for our oceans. One might want to protect it, one might want to fish from it, one might actually get oil and gas, one might want to get minerals from it. And then you have this environmental change that is happening because of climate change, but also because of natural cycles. And they have all the food web interactions. Like if we understand some of that, then that, I think, we start to have a better debate about what levels of certainty do we need and what kind of trade-offs are useful to make. Like it shouldn't just be that kind of like box thinking that we are very, very good in. We need to expand our box.
Yeah. I mean, it reminds me of your of your modeling approach where you need to bring in enough variables to make it relevant to the individual case with the risk of just kind of overwhelming and making it so complex that it's unwieldy, or you might even kind of miss the point.
Yeah.
This was a long conversation, so we've split it into two parts. Tune in next week where we get into some of the